Okay, I see that we are now live on Facebook streaming. So let me begin by saying my name is Daryl Boneth. I'm the editor-in-chief of Marine Mammal Science, a journal of the Society for Marine Mammalogy. Today we'll have our seminar, our monthly seminar. That's part of a series called the Editors Select seminars put on by the Society for Marine Mammalogy. These seminars are chosen from papers that are published in Marine Mammal Science. So before I introduce our speaker today, let me just point out that when you logged on, you probably heard that we are recording. We do record the seminars, including the introduction and questions, and that will be available on the Society for Marine Mammalogy's website subsequently. Just a couple of points. You can put questions anytime you have them in the Q&A. Preferably do not put them in the chat, although we will monitor the chat, so we'll make sure those questions get picked up if somebody does happen to do that. So I guess on that note, let me introduce our speaker today. Our speaker is Dr. Renee Albertson, who has completed a bachelor's degree in chemistry and biochemistry and a Master of Arts in Education, both of those from the Pacific University. Renee subsequently taught high school for a few years and then went on to do a Master of Science degree at Oregon State University. And then she also did a PhD at Oregon State. Both of those degrees were done in conjunction with Dr. Scott Baker, who is also there. The work she did for her master's and PhD involved genetic markers and photo identification techniques to study migratory patterns and abundance of South Pacific humpback whales, and also phylogeographic patterns and social structure of rough-toothed dolphins. Renee subsequently did a postdoctoral degree with Dr. Ari Friedlander, who I don't know if that was at Oregon State, but I think it might have been before he moved on to UC Santa Cruz. Her work with during her postdoc involved humpback whale migration and fine-scale population structure in the Western Antarctic Peninsula. However, today, Renee is going to talk to us about work she's done with colleagues at Oregon State, and we'll focus on gray whales. More specifically, she'll be telling us about the novel use of unoccupied aerial systems to estimate tidal lung volume of foraging adult gray whales, and we'll compare that to tidal volumes estimated from adult gray whales in breeding lagoons. We also have with us today one of actually the lead author on the paper, Dr. James Sumich, and Dr. Sumich will also be available to answer questions at the end of the talk. So I think with that, we'll turn it over to Renee to give her presentation. Thank you very much.
Okay, well, thank you, Daryl, and hello, everyone. Can you guys hear me okay?
Yes.
Okay, great. Thank you for attending this session. My co-authors and I are excited to share some of our work on gray whale physiology with you. I do want to highlight our lead author here, Jim Sumich, in the photo, who began this work literally decades ago. So although I will be going through a lot of equations and showing you a lot of the figures from the paper, I'll also be telling some stories. And gray whales are such an interesting species, one that for a few years we celebrated their population level because it appeared to be near carrying capacity. Now it seems like many species, they face extra challenges in light of climate change. So most of you know this already, but the geneticist in me says I have to briefly discuss the stocks and give a full introduction of the species. So gray whales that we see along the west coast of the US consist of two stocks, and they are actually designated using genetic markers as distinct population segments, or DPSs. There's the Eastern North Pacific DPS that migrates from breeding and calving lagoons around Baja Peninsula or around Baja, Mexico, to foraging grounds either in the Chukchi and Bering Sea near Alaska that you see at the top of the map here circled in red, or the Pacific Coast Feeding Group, or the PCFGs as they're referred to, over on the right side circled in green in the lower latitudes. The second stock, or DPS, includes whales that feed on the western feeding grounds in the Sakhalin Islands and southeast Kamchatka near Russia, shown in blue. Most of those whales migrate to Mexico, and there are whales that possibly migrate to the South China Sea region. So if we zoom in on our map here on the right-hand side, we get a better look at our specific study area. And we were interested in comparing the physiology of foraging and resting whales. The foraging whales are indeed the PCFGs, and our samples were taken in the lower part of this circle. And you can see about where the photo of the foraging whale is, off the coast of Oregon. And these were compared to whales with lower activity levels in the winter breeding lagoons, shown in red, specifically St. Ignacio Lagoon. In the winter lagoons, we collected data from adult females with calves. So here's a photo of a fairly newborn calf. So what did we set out to do? While understanding how a species' metabolic rate varies in different behavioral contexts is useful for quantifying prey requirements and foraging efficiencies of individuals. Field metabolic rates, or FMR, are daily estimates of oxygen consumption, is a function of three factors: lung capacity, breathing rate, and the amount of oxygen extracted from the air. Tidal lung volume is the volume of air exchanged in a breath, which is an important piece of field metabolic rate. And this study describes a novel approach to estimating tidal lung volume, or VT, of actively foraging gray whales, and then compares those estimates to resting adult gray whales in the winter or breeding calving grounds. Before we can get into more of our methods, though, I need to back up a bit. In fact, quite a bit, to January of 1997, and that's where our story begins. This is because some of our main data collection began here on a beach off Los Angeles. So in January in 1997, on this Los Angeles beach in California, a very young gray whale, probably only a few days old, calf stranded, and the mother was nowhere to be found. The calf was extremely dehydrated, and it was decided she would be moved to San Diego SeaWorld for rehabilitation. She was rolled into a sling on a beach, put into the truck, and transferred to SeaWorld. At that time, she weighed 758 kilograms and was 4.2 meters long. JJ, the gray whale calf, was in rehabilitation under veterinary care for about 14 months in captivity at SeaWorld. This created a unique opportunity to collect data on a growing mysticete for the very first time. It allowed for measurements of weight, length, and breathing patterns, all of which were important to consider to calculate tidal lung volume. One of the scientists that was asked to collect these data was Dr. Jim Sumich, the lead author on the paper. So we can fast forward a bit now and talk about some of our primary methodology. So we were interested in these oxygen consumption rates. And oxygen consumption rates, also known as VO2 max, is measured the same way in human athletes or whales. It's just more challenging to measure in whales. And it's dependent on three factors: tidal lung volume, breathing rate, or the ventilation frequency, and the percent of oxygen uptake, or the exchange of oxygen in the inspired air. Although we are currently working on a paper that looks at oxygen consumption rates, including all the variables listed here, we first needed to lay the groundwork and start with tidal lung volume. Therefore, today's topic is focused on tidal lung volume. And you can see in the top left photo where we're measuring tidal lung volume using spirometry, and that is JJ, the young gray whale. So over the 14-month period that JJ was in captivity, these spirometry measurements were collected, along with her length as well. So the other two factors, the breathing rate and the percent oxygen uptake, I will actually discuss towards the end of this presentation. There are two variables that are important for calculating tidal lung volume. That is TEX, or exhalation duration, and body length. The graph on the right shows results for 4 young gray whales and spans a 5-fold range of body masses, from about 1,000 kilograms to just over 6,000 kilograms, which is what JJ was when she was released. What these early data illustrated is that from the individual regressions, which are plotted here for each whale in red, there is no apparent allometric change with increasing body size. The individual regressions are age independent over time. And this allowed us to extrapolate the relationship to adult whales and use this equation to collect data on free-ranging adult whales in the field. So off to the field we went. Because we could use the equation for adult whales, we needed to measure the TEX, or the exhalation duration, of free-ranging adult whales to plug into this equation. We could no longer use spirometry with whales in the wild. So in the lagoon, we used a directional microphone shown here on the left, recording the entire breathing duration upon surfacing. Listening to the audio and viewing the spectrogram shown on the right here, there was a clear distinction between the inhalation and exhalation. The exhalation had a greater acoustic signal. Because we could see where the exhalation stopped and inhalation started, we could determine the time of the exhalation, seen here in seconds in the spectrogram. We could not, however, determine body length, so use the mean body length from data of northbound migrating females with calves of the same cohort. The mean body length that was determined for that year was 12.3 meters for those females. So we were able to collect those data from free-ranging whales, but back in the day, collecting data from free-ranging whales was pretty challenging. And so I just wanted to give you an example of some of the challenges we faced back then. As time went on, thank goodness for technology, because things became a little bit easier. I should also say thank goodness for the GEM Lab and Lee Torres, because Jim and I felt very fortunate to have the GEM Lab within the Marine Mammal Institute at Oregon State University. And not only that, but we had this group of foraging whales, the Pacific Coast Feeding Group, foraging in our own backyard on the Oregon coast. So the drone presented a nice application of data collection for our study for two reasons. One, we could get the duration of the exhale of the TEX value. And two, we could, for the first time, collect data on individual body lengths of whales. So here's an example of what a drone flight over a whale would give us. In order to identify the exhalation duration, we needed to slow the video way down. So here's an example of slowing it down tenfold to get a better idea of what that exhale looked like. Then we realized we needed to look at it frame by frame. However, neither Jim nor I had any experience with video editing. So this was frustrating because we were so close to getting that exhalation data, and we knew we could get the individual body lengths for each of the whales. Enter in Chelsea Harris. Not only was Chelsea our fabulous graduate student, but she happened to have a background in video editing. Needless to say, Jim and I could not believe our luck. So looking at the breath frame by frame, we observed that the nares are elongated during the exhale and visibly flared and widened at the presumed transition point from exhale to inhale. We noted the timestamp of this transition and found no significant difference between the time of observed changes in the appearance of the nares and the actual midpoint times. This meant we could measure the total time the blowhole was open and then divide that by 2 in order to get only the exhale duration. And now we had the final piece of the puzzle for measuring tidal lung volume for individual whales. So here then is some summary data from the exhalations of the foraging whales and the fasting females. And if you focus in on the highlighted part of the tables show the mean TX or the exhalation data, the foraging whales in the top table, and the fasting whales or the lagoon whales in the bottom table. The exhalation is slightly longer for the foraging whales, which we would expect given they're working harder. The graph shows the relationship between the exhale duration and the body length from individual whales in the foraging grounds. As shown from looking at the slope, as the length of the whale increased, so did the exhalation duration. These mean VT values for each individual foraging whale were then calculated using the equation that included the length of the whale and are plotted against their calculated mass from this second equation, which is the graph you see here. We see the same relationship then with body mass as we did with body length. As the body mass increased, so did the tidal lung volume. And this brings us to our first summary point of gray whales. Where does this put gray whales comparatively to other cetaceans for tidal lung volume? Well, there are no tidal lung volume body mass ratios of mysticetes at any age available for comparison. But when compared to odontocetes, we can see kind of where they fit in. So here in the graph, there's the 1%, 3%, and 5% regression lines. The 1% is the one farthest on the right, that's the dotted line, and that is of terrestrial animals. The middle is the 3%, the dashed line, where you see where the gray whales fit in. And also around that size are our medium odontocetes, like Tursiops towards the bottom, and then beluga whales. And then that top 5% is where we see the larger odontocetes, killer whales, and short-finned pilot whales. So then, now that we have that first variable and critical component of that oxygen consumption rate, our work is now looking at the other two variables to measure metabolic rate and aerobic dive limits of gray whales. And as I said, these last few slides are indeed preliminary because this work has been submitted, but it is under review. But we thought it would be fun to share some of this work with you and look at some of these possible insights. So formally, just a little definition of aerobic dive limit, as defined by Kooiman, is the diving or breath holding duration threshold beyond which there is a net increase in lactate production. And ADL was originally determined by blood lactate measurements based on Kooiman's classic studies of the Weddell seal. Determining ADL from blood lactate levels requires repeated sequential blood samples during and immediately after a monitored dive. So with few exceptions, studies of cetacean ADL have been focused on odontocetes. So I showed you a video earlier of how challenging it could be to collect data from free-ranging whales, and therefore for unrestrained whales that dive repeatedly, taking blood samples was not an option for us. But the good news is ADL can be calculated using total oxygen stores, which was part of our original equation with the tidal lung volume, and then the rate of oxygen consumption. So this that I showed earlier showed for oxygen consumption rates, we found the tidal lung volume. The others, the breathing rate was actually a little bit easier because that's looking at the breathing rate of the whales. And the last one is the percent oxygen uptake. So by easier, I mean the breathing frequency or the breathing rate is easier to collect in the field because this can be done by observation. So we had the tidal lung volume, and the breathing frequencies could be easily done by observation in the field. And the percent oxygen uptake data that we have was from JJ. Next, we need to calculate oxygen stores. And all of these features are partly dependent on body size. So this is for a standard 12.2 meter long whale, and that's about 18,000 kilos. And this is assuming a lung volume that's about 80 liters, blood volume from the hemoglobin 270, and the muscle from the myoglobin about 100. But here's another way of looking at it that's sort of interesting with gray whales. Gray whale blood stores account for about 60% of their totals, with significantly less in their muscle stores when compared to other mysticetes. So looking at the graph on the right, the comparison of gray whale muscle oxygen stores to fin whale and blue whales is interesting. Gray whale muscle oxygen stores are less than half the mass relative oxygen stores reported for fin and blue whales. So if we plug those numbers into our equation to get our aerobic dive limit, we get an aerobic dive limit for gray whales of about 9 minutes. But what's interesting is if we plot that in a histogram, the histogram shows this in more detail, that only a very small portion of the dives is approaching that aerobic dive limit. So if you look at where the arrow is for the ADL, it's at 540 seconds or 9 minutes there along the x-axis. You can see there's very, very few dives that are actually to that limit. In fact, it's only 0.06% of those 9,000 dives that are exceeding that aerobic dive limit. And we found that kind of surprising. So we looked at that compared to extended dive times. And we define extended dive times for gray whales as any dive that's over 1 minute. So the figure shows that for gray whales, that mean aerobic dive limit is 4 to 5 times greater than the mean extended dive time. So this in itself is actually not too surprising. Other studies of mysticetes, specifically blue whales and fin whales, have shown similar results with their predicted ADLs about 4 to 5 times greater as well. But what is surprising or worth pondering from these results is that unlike many phocids and some odontocetes who regularly approach their aerobic dive limits, gray whales seldom approach their physiological dive limit. And perhaps it's been said that may be under little selective pressure for developing physiological adaptations, such as high myoglobin content, that would prolong dive duration. So again, they seldom are approaching their actual physiological dive limit. So why the large difference between the aerobic dive limit and the extended dive time? We have no idea. But in known high predation areas, such as Monterey Bay and the Alaska Peninsula, northbound adult female gray whales and their calves often exhibit stealthy or cryptic behaviors, including long duration dives greater than 10 minutes, gentle surfacing, and slow, quiet expiration. It is possible that maybe ADL is sufficient to allow for infrequent yet critical prolonged aerobic dives enhance a gray whale's capability to resist or evade a killer whale's attack. And this has been an important selective factor in the evolution of gray whales' aerobic dive limit. And so just to circle back to the beginning and why we set out to do this in the first place, gray whales have actually experienced a 46% reduction in population size in just 8 years. And with climate-driven oceanographic conditions negatively impacting gray whale prey availability. And as scientists, I think we are united in our work assisting policymakers in future management decisions and ultimately the conservation of species. So our hope is these new tools, especially the ability of drones to simultaneously establish body lengths and monitor breathing rates of individual whales, provide robust estimates of gray whale tidal lung volumes and field metabolic rates to better understand resource requirements. And with that, we would like to acknowledge all the many organizations that contributed to this work. And we will take any questions.
Thank you, Renee, for that very interesting presentation. I guess we do have at least one question that has popped up here, so let's go ahead and turn to questions from our viewers.
It's more of a comment. It says gray whale prey species vary according to foraging area.
Yeah, agreed. And I think, you know, one of the things that has come up is that our work has looked only, you know, at the Pacific Coast Feeding Group off of Oregon in the foraging areas. And it would be really interesting, I think, to collect some of these data up in, you know, some of the other foraging areas as well.
I have a question. Because you just mentioned the population decline and that this technology is quite new. So is the goal to monitor any changes over time in the future to see if it's changing with any changes in the environment or prey availability?
This work, I think, can be a piece of that. It's not, you know, I think that is a part of many collaborations. Jim, I don't know if you want to chime in on that.
Well, this is kind of an ongoing study, so certainly if anything interesting pops up where we can do a time over time comparison, we certainly would. But because there is a change of individuals involved in this area that where the study has occurred, it would be pretty difficult to separate that out from any signal that was just time related.
Yeah, thank you. And there's a new question coming in saying, off the coast of Oregon, what is the primary prey species observed?
That's actually, there's quite a few actually that we weren't, I mean, mysids, I think, were the primary prey species we were thinking originally. But we're finding that there's a few more now.
Yeah, mysid shrimp are probably the primary one, but gray whales are a little bit opportunistic. So, you know, in times of heavy spawning for other kinds of crustaceans, those larval stages are also potential prey and certainly are being utilized.
Yeah, like porcelain crab larvae, for example, is one. There was quite a few. We were surprised that they were so opportunistic, I guess.
Thank you. And then one more question here. Have you found any differences in tidal volumes between male and female gray whales?
The easy answer is no, because it seems to be more size related than gender related. And sometimes it's difficult to determine sex of individual subjects. Females tend to be quite a bit easier, especially if they're in the presence of a calf. But it requires not only longer term observations, but also tissue sampling and genetic sexing. And we really haven't looked at that. It might be, if we had a larger sample size, I think it would be worth going back and taking a hard look at.
So not much sexual dimorphism then in terms of size differences between the sexes?
I'm sorry, I didn't hear that.
So there's no size difference between the sexes?
There's a size difference between the sexes at maximum size.
Right.
But over that range of growth, you can't use size by itself to determine sex.
Mm-hmm. Thank you. One more question about prey species. In the Bering Sea, are bottom-dwelling worms the primary prey species, perchance?
No, they're primarily bottom-dwelling amphipods. tube-dwelling amphipods in the genus Amplesca. And we do have some foraging on amphipods here along the Oregon coast, but they're not the primary one like they are in the north.
Thank you. And I'll just read this out. Hi there. I would like to just ask if these measurements can be used to look at carbon dioxide exhaled of these animals. For example, with papers looking at sperm whales as a carbon sink, multiple methods have been used to determine the balance of resource intake and excretion. I hope this makes sense.
I'm a little fuzzy on the actual question.
Can the measurements be used to look at carbon dioxide exhaled? So I'm guessing the amount of carbon dioxide that is being exhaled. And in a
Certainly that
volume
would
or...
require sampling exhaled air. And in these kinds of field situations, it's essentially impossible to do. I mean, we've been fortunate to be able to do some with JJ in a controlled situation, and to a very limited extent in winter lagoons with animals that participate or at least allow that sort of contact. But in feeding situations, no.
All right. And then next one is data from GG, the first gray held for 1 year in SeaWorld. Any data from Jim's earlier 1980s exhalation studies at LSI? Starting with
GG, some of the data that we have included in this paper is from GG back in the early 70s. And she was released at about almost exactly the same age as JJ was 25 years later. But a lot more sampling was done with JJ than had been accomplished with GG, the earlier one. But we have...
Yes, yeah, I really enjoyed all the methods background that you showed. That was super interesting. Thank you, Renee. Good work there, Jim. And there's another question here. So if the gray whale population is dropping, is part of the decline due to starvation?
We wish we knew, but we certainly do think that it's resource related, food resource related. And it's not just starvation, but a reduction in successful reproductive rates. Both of those are contributing to a decline here in almost the last decade.
Yeah, and I think looking at, Lee Torres has been doing this study on body condition, and that's certainly been a factor, is seeing more of what we would refer to skinny whales in that regard in the last 5 years, wouldn't you say, Jim?
Yes, and it's sometimes difficult to really determine cause of death, or at least relate cause of death to starvation, because animals short on resources in weakened conditions may very well succumb to other sorts of health issues rather than directly to starvation. But the lack of food resources could still be the major contributing factor there.
So because you mentioned the skinny whales, are you using the drone footage on other projects to determine maybe the weight or changes in the weight distributions of the whales?
Well, Lee is one of her main studies with the drone footage is looking at body condition of gray whales. I can't remember what year she started that, but I feel like it was 2018 or 2017.
Yeah, it's been at least that long, yeah.
That's all the questions I can see for now. Feel free to ask more.
So I have a question I might ask, and this, I don't know, maybe it's my ignorance here, but so with regard to the hypothesis of predation pressure being a possible explanation for aerobic dive limit differences in gray whales and other mysticetes. Are there gray whale populations that are quite different in sort of killer whale predation pressure that you might be able to look at the differences between populations of gray whales? Does that make sense?
Yeah, I don't think we have enough data to look at that specifically at this point.
Well, and part of the complicator there is that at least for the predation that occurs along the West Coast, all of these gray whale populations or subpopulations, the Westerns, the Eastern North Pacific population and the PCFG whales, all of them, or at least members of those group, all three of those groups, migrate through this corridor along the West Coast. So the predatory pressure could be applied to individuals of any one of those three groups.
Okay. So if we have no other questions, maybe I have one more I'll ask, and it relates to the figure where you showed number of odontocetes in relation to the gray whale, and I'm trying to remember what the axes were. But
it was tidal volume versus body mass.
Okay, and what struck me was the comment about the larger odontocetes. And yeah, so the larger odontocetes are falling up here, and I'm surprised if in fact size is a major driving factor, that then gray whales fall down here rather than up where these larger odontocetes. Does that make any sense or not?
Yeah, it does, and it may be that what we've done is to switch taxonomic groups, and that the pattern that we see with the odontocetes is showing something different than what we would see with mysticetes, particularly if we had more information than this one species and two areas of sampling.
Yep, makes sense.
Because in terms of the other things that we're looking at, in terms of oxygen storage capacity, gray whales seem to be substantially different than either odontocetes or especially phocids, but probably somewhat different than other mysticetes as well. That may reflect the kind of foraging, the kind of diving patterns that they have. I mean, at least along the Oregon coast here, most of the foraging is done in very shallow water. So depth really doesn't become a factor in terms of characterizing these dives. A lot of the foraging is, less than a body length in water depth compared to what you would see with larger mysticetes like humpbacks or blues.
Okay, thank you.
There's a comment in the Q&A, first of all, from James Hadnell saying, given the number of unknowns regarding the drop in grey whale population, I'd be cautious about linking the population drop to climate change. I don't know if you want to respond to that. You can. And I've also got a question about the aerobic dive limits. I'll start with the comment and then read the question last. It says, with mysticetes especially, the body size is an important factor dictating the CADL. Smaller marine mammals that must dive for extended periods of time tend to have enhanced myoglobin, etc. And the question is, might the difference in CADL and actual dive times be a result of the feeding strategy not requiring these longer dive times?
I think that's a very good question, and it's something that we've been asking ourselves. I think that what would really be helpful is a fairly effective study looking at the ventilatory patterns of northbound lactating females. This is a big gap in our understanding of what these ventilatory patterns. We've got a lot of information on southbound migrations. We've got quite a bit of information on foraging, at least in the PCFG area. What we don't have really very much information at all, except for anecdotal observations, is what's happening with the northbound adult females in the company of their calves.
Excellent. Very engaging today. Love it. That's all the questions I can see for now.
Okay, I guess I'm not sure I have any further questions. So Sophia, unless you do or Teresa has any questions, I will thank Renee for a very interesting presentation, and thank you also, Jim, for participating. And hopefully we look forward to future work being submitted to Marine Mammal Science.
Very good.
Yeah, well, thank you for the invite. We appreciate it.
Great. Well, thanks much,
Oh.
and stay tuned for another seminar next month.
Sounds
With
good.
that, I guess we'll say good night on this end. I guess it's still daytime for you guys.
Yes, it is.
Yes.
Yeah,
Thank you all for your efforts here.
you're welcome. Thank you. All right. Good night.
Good night.
Thank
Thanks,
you all.
Renee. Yeah, that was a great presentation. I followed more than I might have expected, so you did a great job.
Oh, thank you.
And thanks, Renee, for stepping up and doing this.
Yeah, no problem. It was a good team effort.
Good. Well, again, look forward to more papers coming out of your lab.
Sounds good.
Okay.
Thanks, Cheryl.
Yeah, you're welcome. Bye-bye.
Bye.